It's an amazing shot.
No. Hello, Stephen.
Hello, good afternoon.
Hi, Rikia.
Hi.
Hey, Steve.
Hey, Thomas, good to see you.
Good to see you too. I stopped by a few weeks ago, but you were in clinic.
Oh,
I think you're at the clinic, and then I just the army has been keeping me busy, so I haven't been able to make it back. But good
Yep.
to see you on this. So let's see. We'll wait one more minute, let everybody else trickle in on this first Thursday of November. Okay, let's get started. Good afternoon, everybody. Welcome to our HiPAM weekly Thursday meeting, first Thursday of November. So, man, October really went by quickly. Hopefully everybody had a safe Halloween, wore masks if you did go trick-or-treating or did the drive-through trick-or-treating. I think I just stayed home. But let's get started. So, from last week Thursday to this week Thursday, understandably, based on if you're just reading the news at all, right? It was expected that the 7-day case average increased from last week Thursday's around 74,500 to a little over 86,000, and not surprising because over the weekend we had over 100,000 daily infections across the nation, so that's obviously going to drive up the averages. Focusing on our local snapshot, these are our numbers from last week in terms of our 7-day averages of new COVID cases, 87, increase to 92 for this week. I think we pulled this yesterday. Hospitalizations did go down as did fatalities. I apologize. I tried to look this up right before the meeting, but based on Hawaii Data Collaborative's website, it said that yesterday's 7-day case average was 156. That might be an error based on what was shown here. I believe it's more likely to be 92 in terms of our 7-day average of new COVID cases. I know we had 156 yesterday, but for our collaborators from HDC, if you wouldn't mind checking that on your end, because that's a huge jump from 62, but it makes sense to go from 62 to about 92 from last week to this week. No major change in the 7-day average of new deaths, obviously, because deaths is fluctuate so much that it's really hard to take anything away, minus the fact that it's good that we're trying to keep on trending flat or even downward, especially for this data point. New hospitalizations, 7-day average continues to move downward, and once again, keeping with this great trend of ICU hospitalizations. Red is the number of COVID ICU and that once again it is continuing to trend downward over the weeks. If you look at overall ICU utilization, it's not too bad, which is a good. That means that we're utilizing it for normal capacity. Looking at percent positivity, we were trending down as a state, but once again, based on some of the higher numbers the past few days, we're seeing an uptick, 2% from last week to 2.1% this week. So something to keep in mind, we're still under that 2.5%, at least for Honolulu County, to go from tier 2 to tier 3. And then our testing turnaround timeframe, pretty consistent in terms of the overall magnitude of tests that were turned around within 24 hours. However, if you look more of a proportion, a slight decrease, but that's to be expected potentially as we're doing more of that surveillance testing. I'm not sure how much of that second post-test surveillance study is contributing to these numbers. Once again, as we had opened up in 15 October for our Trans-Pacific travel, we know that these 3 states really contribute heavily to a lot of our tourists and returning residents. We have a lot of family members that live in these states, so keeping an eye on those trends potentially as that could impact infections locally. They're all trending up. Second straight week, all three states, all 3 of these states have continued to trend up. We added this last week, and we'll continue to look at this as there's more and more talk about opening up to Japan. It was trending up last week, slight tick downward, but once again, just continuing to monitor this on a week-to-week basis and see where that goes for us. This is a new slide. If you looked at HiPAM's website over the weekend, we did upgrade thanks to Levi's great work. Levi, I'm gonna mute myself and let you explain this this quad chart because you better than anybody else could provide some guidance on this.
For sure. So this is pulled directly off the HiPAM site, just adding a couple kind of numbers for additional clarity here. And this is all the numbers that we're forecasting for the 7-day case average, deaths, hospital beds in use, and ICU beds in use. I would like to highlight that we're kind of at another one of those unpredictable points in time, just because as you can see this really big spike in the last couple days from where we were from a 7-day average point of view. Typically what happens is we might see a big spike and it'll kind of level off for at least a little bit. So it's not necessarily going to continue in this perfectly kind of straight up trajectory, which is why I kind of have it flattening out a bit. This is probably, I would say, subject to change, within the next couple of days. It could come down or go up relatively dramatically just based on the way the trend is. It's always hard within the first couple of days here. So just note, as we're looking here, we are projecting an upward trend, both expected and kind of a worst case scenario, but we're still kind of going to be looking at that really closely over the next few days. And as a reminder, this is a 2-week forecast. So when we're looking at things like deaths, we're not going to really see a really big divergence between both of our forecasts. We still are predicting, another 12 deaths or so. This could and will continue to go up if we do continue to see this increase. After these 14 days, that's typically when we kind of see the deaths start to continue to rise with it. So just note that this is really a lagging measure here. And when we're looking at the hospital beds and ICUs, we're in a way better place than we were in the last couple months. Like we are still on the rise, but we have a lot more leeway as of at least right now, especially when we're looking at ICU beds, forecasting to still be around 10, even with this uptick here, because even though this line is steep for the average cases, it's still relatively low, all things considered. So we're still in an okay place, but again, it kind of could go either way just based on how the next few days play out.
Thanks, Levi, for that. A couple of things to add to what Levi said. This is where, we're trying to be as scientifically based and data driven as possible. And we have good data from DOH. And this was a discussion that we had last week with Dr. Berman. There's so much to the nuance of understanding the spread of infection for COVID. And we understand there's a lot of divergence when it comes to age as one of the major predictors of morbidity and mortality for COVID. And so, if there's more clusters or other types of data that can help us better understand the transmission, then what that does is it helps us kind of better explain, that increase that Levi mentioned on the cases, but why we might not see that much of an increase in hospitalizations and fatalities, just because once again, it depends what the age breakdown is for our infection. So once again, we're working with the information that we have, which has improved tremendously, but understanding the nuances with COVID, that it limits our ability for some of our forecasting. So 2 articles, there's always so many, but 2 that I wanted to highlight for this week for multiple purposes. There's facial masking for COVID-19, potential for variolation as we await a vaccine. That's more to highlight a higher strategic level issue that we're continuing to experience. And the other one is outcomes of contact tracing in San Francisco, test and trace during shelter in place. So this first one, hopefully everybody should be familiar with New England Journal of Medicine, one of the premier academic scientific journals. They published, I believe, a few days ago, a perspective on 2 MDs. Kind of their point was to say that maybe use face masks for variolation, which and I apologize to the MDs in the room, because this is definitely a little out of my scope, but to the best of my understanding, kind of using the mask to reduce the dose of potential COVID that you might inhale, to the point where you could get some sort of immunity without getting so much of the virus that you're gonna become fatal. But that was kind of what they were pushing in this article. And so there was a lot of responses that were published to this particular article. A lot of limitations. This was only a hypothesis. There was nothing based that these authors based this perspective article on. Obviously, we all know there's too many unknowns. We don't even know if there is, or what the dose response curve is for COVID particularly. And they don't really define face covering. And we know there's a million different types of face coverings, you can buy on Amazon. And so the big picture takeaway is, first of all, the fact that New England Journal published this, it gets anything that's published in this article, in this journal, gets a lot of publicity. And what may start as a misunderstanding can easily turn into misinformation. And so this is a perfect example of, it's an interesting hypothesis for sure, but a lot of times when people read it, they're like, oh, this, because it's published by clinicians or researchers and it's published in a high impact journal, there's something to this and maybe we should try this out. And so, and the reason why I brought it up is, as HiPAM and as we're leaders in our field and in the community, it's just so easy for anybody with this, especially with social media, to take kind of what we're talking about, misuse it, and then we can see the massive impact, negative impact that it could have. So this is just a perfect example at a national and an international level of potential misunderstanding leading to misinformation. It's still a worthwhile read, but there's a lot of limitations to this particular article. The other one I wanted to highlight, this was published, I apologize, I did not put the journal. Let's see if I can go back. This was in JAMA, so once again, another high impact journal. To a bunch of public health researchers within San Francisco Department of Health. They actually did some pretty good analysis, in-depth analysis into their contact tracing. And what they found was that within San Francisco's contact tracing program, they identified, these are 2 of the major highlights that I found. More than 80% of cases and contacts identified were secondary cases within a median time frame of 6 days. And then they actually broke down some of the ethnic breakdowns as well. The other significant finding was because 90% of secondary cases were household contacts, pre-symptomatic transmission could have occurred, such that infected contacts may have already transmitted the virus by the time they're notified, once again confirming what we pretty much assumed to occur. But it's always good to get hard contact tracing information and data to support kind of our hypothesis. So twofold. One from the data standpoint, this confirms a lot of what we understand for contact tracing information, but also hopefully this is something we could potentially see coming out from our DOH as they get more and more capacity to do that, or if they have the ability to share some of that information with the outside partners. This is a great addition to the knowledge base specifically for San Francisco, and hopefully Hawaii can get to that point in the near future as well. So before I open it up, just wanted to do a quick push for COVID POW. They have a town hall. The Facebook link is here. We'll make sure these slides are available after this meeting. Here's the moderator, the panelists, but once again, COVID POW is another community partner focusing on really good messaging that's based on data-driven facts and scientific information to really help the community in Hawaii better understand COVID, all things COVID. So I'm just putting that there. With that, I will stop sharing just for a moment and open up the floor for discussions about the data points that we've seen, the trends, the changes to our webpage, some of the modeling questions Levi can answer, or the two articles that were shared before we get to the deeper dive by Margarita and her fabulous students.
I would just say that when we find that contact tracing finds a large percentage of household contacts, I would take that with a grain of salt, because it's probably easier to find household contacts. So people might meet casually with somebody, but they cannot remember, they don't know who that person is. So I think that's probably a biased number. I'm just, I don't know how to establish the true percentage.
No, you're absolutely right. It's fun. I mean, it's especially with the nature of COVID, where you can have so much of the transmission be asymptomatic or pre-symptomatic. It makes it extremely difficult to nail down all the contacts that, excluding the household contacts, because it's so hard for people to first recall it, and then, if they're asymptomatic, they're not even gonna know, or the other people who might get it might not know who was that same person at the same event. So it's just extremely difficult, regardless of how hard or how well the contact tracing capacities are, especially if we're at a higher baseline of transmission. So that's a really good point. No other questions? You guys are making it easy for me today.
Okay, maybe one more comment if nobody wants to say.
I spoke too soon.
Sorry, just I kind of expected that the opening of the 50s would start showing up around early November, and so it's only a hypothesis, but this spike we are seeing actually could be the sign, because of the kind of delayed nature of the daily cases compared to this time scale is 2 weeks, so it's like the first time it could show is like in 2 weeks time, and it did show. So it's plausible to think, I think that's the effect of travel, and we'll see, we should watch in the next few days what happens, as Levi said.
No, I absolutely agree. It's fun with what we know about the incubation period for, whoever brings it in, or, from that 15 October, it's about this time. I think most people who are tracking how COVID's transmitted, this is, I think, the week that we're really looking at to see if there's a spike, at least, or this weekend, next week. So I think we're all keeping our eyes peeled.
Thomas.
Yes, sir.
Since this John opened the door, as we lawyers are wont to say, the most obvious way to tell or to confirm whether it's the spike is due to travel is to have perfect contact tracing. And we know we'll never have that, but to the extent that the contact tracing is functioning, we should we should know some of that. But based on what the DOH has shared to date, it's not likely that at least anybody in the public is going to know the extent of maybe perhaps what the DOH knows. The other part of that is anybody that's been on this call for any amount of time knows that I've referenced the UHERO June 9th brief, which analyzed what I'll call the pretest, the single pretest program for inbound travelers. They'll also know that UHERO published an extension of that to analyze the impact of opening to Japanese travelers. And in the time I've been watching this, I've noticed that I've learned a little bit about the dynamics between the various members of Hawaii's public health community. And it's shockingly, it's much more complex than I would have thought at the beginning. I've used UHERO's briefs as the quintessential credibility, and that things, lawyers look at things in terms of authority. That would, to me, would be like controlling authority, which means the court is bound to follow it. It has that kind of credibility. And then you get down to persuasive authority where, well, maybe you don't have to follow it, but you better have a good reason not to. One of the things I've learned is that my putting them on that pedestal may have been a little short-sighted. So I'm wondering, what HiPAM's reaction is to, we were told at the beginning that we were going to get, the mayors at least, were going to get details of this, what I'll call the surveillance program. The only detail we've got so far is a press release issued a couple of days ago that gave some very vague numbers and nothing about the methodology or anything else. But one way, if we thought that the spike in cases was due to travelers, then I would think that the viability of that testing program would be something that would require some more scrutiny to see if it doesn't need to be challenged or if it doesn't need to be bolstered. And yet I don't see HiPAM doing that, but I'm kind of wondering if there's anyone in the group that has any thoughts about what HiPAM's role might be in either, analyzing that program and saying, yeah, we signed on to this. We think it's credible. We think it doesn't leave any cracks, or challenging it and saying, and whether that's done on a public or private basis is a whole separate question. But there's certainly a lot of questions out there. So I'll just throw that out and let people stew on it a little bit.
Thanks for that, Jim. I will defer to Dr. Berryman and maybe Dr. Henke.
I would comment if I could on a couple things that Jim said. One was of course, we're never going to have perfect contact tracing. So, we would all like that. We are, though, in a better situation than we were over the summer when our cases were at a peak and our staffing was low. So in conversation with Dr. Kemble, we're right kind of on that cusp of actual containment and mitigation mode, with mitigation mode being more you pay most attention to the higher priority, and containment being you do your best to talk to every case. So if we don't see continuing escalation, we should be able to stay in that containment mode. But it won't be perfect. But where we may see the impact of travel initially is in the epi curve that I haven't looked at for a little while online, but the epi curve, the classic version, I guess I would say, that's red and blue, that shows the travel cases in dark blue, the travel associated in light blue, and the community acquired in red. So anytime we identify a case in a traveler, that's the dark blue category, and the light blue is anybody who's clearly epi-linked to that traveler. So it doesn't go to all the generations of transmission that might be there, but that category has been very small statewide until recently. So that, I think, is where we can see those numbers and keep an eye on any trends there. I will say that on Kauai, we've had since the 15th of October, we've had 13 cases, and all of them have been travel related, and that's many more cases than we were seeing, up until this point. And a significant number of those have been people who had a pretest, arrived with that test pending, and got a positive test after arriving here, which poses another whole range of challenges, because that means that people around those travelers on their flights were exposed and also need to be quarantined here. So it's complicated.
Steve, do you have anything to add or comment on this?
Yeah, thanks. And I know I'm new to the group, so for those who I haven't met, I'm Steve Hankins, and I'm a family medicine physician, but also have a master's in public health and done emergency response and pandemic work for a number of years, and have been working at the state with ESF8 since about the same time Dr. Lee joined the group, maybe a little bit before, since I was successful in recruiting him to ESF8 initially. So, I think these are all very good comments and very important issues to keep in mind. I think it is important to understand, again, context and the interface between this disease entity and the community of individuals that it's interacting with creates that very dynamic variability and mathematical chaos that we see played out over and over again over the distribution of curves that we see when we pull up different geographic jurisdictions. Kauai is certainly one of the few fortunate places on the planet that, because of its combination of geographic isolation and its very robust posture in terms of limiting importation of disease, has such a low case rate within the community, and it's also much more geographically less dense and in terms of population. So the populational transmission dynamics are quite different there than they would be in urban Honolulu, for example. So you're clearly going to see the impact of importation of disease much more early and to a much larger disproportionate degree, I think, than perhaps what you would be seeing, for example, here in Honolulu. And we need to keep in mind that with the reopening of travel is the return of much social interchange in terms of work for those that work in the travel industry. If you travel on the roads, you'll notice that there's been much more traffic. There's much more, there are many workers who are now sitting down at break tables that have been apart for months and months. And so, again, there are all of these other aspects of the reopening of the economy and return to that travel is a part of. That's not to say that we shouldn't, you're absolutely right, James, be watching very closely what we can ferret out about the impact of the travel component of it. But I think it's going to take a while before that data is very clear, particularly in the more the places where we do have higher levels of community transmission at baseline. So those are one things. In a more general sense, I did want to comment, one of the things that I would note from an overall impact and posture of how we think about responding is that while we have been at this nice lower plateau for a little bit now, as Levi was pointing out, it's significantly higher than where we were before the last curve, the last spike in numbers. So our window between where we currently are in terms of capacities across the system and where that ceiling is of what we can handle is narrower. And the other thing I would say is just looking globally at this third wave, if you will, is that it is proportionately larger in most places, certainly in Europe and across the United States in general, in terms of daily case numbers and so forth. So I do think there's reasons to be concerned about what the coming months may bring.
Thanks so much, Steve, Dr. Hankins. I love the great discussions that we've had, but I really want to give Ethan and Claire, I don't know if you're presenting this week, but Marguerite has some fabulous students that prepared really good slides that I'm really excited to hear because they're way smarter than I am. I can only speak for myself when it comes to this topic. So I'm going to go ahead and share my screen again. Ethan, I'm super not tech savvy. I will be your Vanna. So I will control, I'll control the slides and you tell me where to point and I'll point, I'll just be silent and you can take away. Unless do you have your slides on your computer? I can give you access, Ethan, you can share the screen.
Yeah, I have my slides up.
Okay, perfect.
Okay, we can have access. That'd be awesome.
Of course,
Thank you.
that's super easy. Just give me 1 second.
Okay, so yeah, so thank you very much. I'm super excited. Claire's going to talk next week, but I just want to say that and so our lab studies biodiversity actually. And once the COVID pandemic hit, we all wanted to try to contribute. And so Ethan and Claire both got really excited about potential to use genomics to study SARS-CoV-2. And so Ethan's going to talk about using genomic surveillance and how we could help Hawaii. And Ethan's a master's student. And so, yeah, thank you. Take it away, Ethan.
Awesome. Thank you for that introduction, Dr. Butler. Do I have host privileges by any chance?
I'm working on that right now. Apologies. Rukia, can you give Ethan access, please? I apologize. You're still the host, so I don't have that ability to.
Okay, awesome.
Okay, there we go. Does it work for you?
Yes, I believe so. Okay. Can everyone see the slides? Perfect. Can everyone see? Awesome. And one more thing. I just want to make sure this works. Can everyone see the laser pointer? Okay. Perfect. So thank you for that introduction, Dr. Butler. My name is Ethan, and I will be talking about tracing SARS-CoV-2 with genetics, specifically their genomes. So just to give a little update on what's been happening. So as of yesterday, internationally, we've hit 47.6 million cases. And within the US, yesterday, we were at 9.4 million cases. And we're currently observing a third wave of infection sweeping the continental United States. Internationally, the UK just shut down recently. And contact tracing efforts, as good as they are in the process of being overwhelmed, especially with this recent surge in cases. So the natural question to ask from this is, can we use genetics to trace the spread of the virus? And a little background on the genetics of the virus so far. So currently, the SARS-CoV-2 genome has been sequenced and annotated, which is really important because we have been able to identify functional regions in the virus and what they correspond to when they make the proteins that they need. We've identified that the genome is approximately 30,000 nucleotides long, and this genome mutates at about two mutations per month. And in the grand scheme of things, this roughly translates to about 1 mutation per host transmission, which is really cool. because we can actually use these mutations got from different samples and infer the pattern of spread between individuals. So to give you an idea of what I'm talking about, say we have patients A, B, and C. They have all been confirmed positive patients, but we don't know how they're related. Using the mutations present in the genome, we can actually identify who has that unique mutation, or who has this, the virus that has this unique mutations, and trace it back to see the pattern of spread. And studies like this have actually been conducted before. They're called phylogenetic analyses. And what phylogenetic analyses do is look at the evolution and the pattern of spread of these viruses through time. And this actually goes well beyond just contact tracing methods, because instead of just understanding the short-term spread of the virus, we can actually look at the spread of the virus across space and time across its entire family history. And this was actually done to study a recent yellow fever outbreak in South America. Going into the study, the researchers knew that there were 2 distinct strains that were present, but didn't know if the pattern of spread was different. So what they were able to infer from using the genomes of this yellow fever virus is that the old lineage was actually introduced into Brazil specifically in the early 1900s. And then what they were able to find is the actual pattern of geographic spread. So what they were able to find is that the initial introduction was into northern Brazil, and then from here, it actually radiated out from Brazil into Venezuela and the countries along the northern border of Brazil, and also down to the different regions of Brazil. And this was very, very specific to the old lineage that occurred between 1946 and 1992. However, with the second lineage, they found a very, very different pattern of dispersal. What they actually found was that the second lineage, the modern lineage as they called it, was introduced in Venezuela, in Caracas, and then through jumps between neighboring countries, as well as a lot of long distance dispersal events, saw that it kind of stepped down the South American continent, eventually making its way to the bottom. And remember, this is the same virus, just 2 different lineages, and its spread happening at 2 different times. And the cool thing about this is that studies like this have been happening, and studies like this have also been starting to happen for SARS-CoV-2 as well. Oh, there we go. So here's actually one of the earliest phylogenies or family trees of SARS-CoV-2. This was generated in March and was published in June. And what you can actually see is the timing and dispersal of the virus. So I'll take you through it step by step. So the first genomes that were sequenced for SARS-CoV-2 were sequenced in late December, early January, and they were primarily, or they're all sequenced in China. And based on this location to the root of the family tree, they were able to infer that the virus started in China. And then what we notice is that as we fast forward in time, so starting in around mid-January to March, it had spread all over the world. Each of these colors represent a different country that you see over here. And what's even cooler about these types of analysis is we can infer the timing of certain dates. So if we look at the oldest node over here, sorry, I don't know if that came up, what we'll see is that the basal split of this tree implies that the first jump to humans occurred around late October, November timeframe. And we can see that around January, we can see the first transmission or dispersal from China to the United States over here, specifically in Washington. We can then see a dispersal from China to Europe over here that occurred in mid-January. And finally, yet another introduction into the United States that occurred in mid-February right over here. And this one was introduced to California. From these analysis, we can also take a look at inferential statistics like the effective reproduction number, or RE. And based on the sampling through time, we were actually able to observe a dynamic RE that changes based on potentially actions that governments are taking to restrict the movement of people in and out of the country and within the country as well, like lockdowns. Then the really cool thing about these is the more genetic information that becomes available, the more powerful these analyses become. And you can actually start looking at populations of virus on a finer scale. Here's a study that was conducted very recently on the SARS-CoV-2 cases that happened in Brazil. And they're actually able to first identify the timing of introductions into these countries, which they put around mid-February right here. And additionally, not only were they able to identify the timings, but they were able to identify the distinct lineages that were introduced. And interestingly enough, there were 3 distinct lineages that were dominating the population right here. And we can see that they were all actually introduced around the same time. Furthermore, we can also fine-tune our understanding of the geographic spread of the virus within a region. And this is where it gets really cool. Because what we can see in Brazil is that they've identified 2 phases of the pandemic. So the first phase of the pandemic that they identified occurred between late February to mid-March. And what they found is that the majority of transmission that occurred within Brazil was primarily local. And it was really condensed in the metropolitan urban areas with very few introductions happening or dispersals happening to the outer regions. However, in the second phase of the epidemic that occurred between March 21st and April 15, they found a lot more long distance dispersal events. And what they actually found is that a lot of these long distance dispersal events brought the virus from the fringes of Brazil back into urban centers. Which was, I mean, this information is amazing because we can actually study how the virus spreads within a region in pretty close to real time. Yeah. And we could actually take this sort of analyses and apply it to the US and actually specifically of Hawaii, specifically to Hawaii. And we could answer the following questions. We could definitely identify the origin of the samples and strains that we have currently in Hawaii. And we could very easily distinguish between imported strains as well as strains that were spread primarily through community transmission. We can infer the transmission lineages, so we can identify local clusters of who was infected and who got it from who. We could also look at the effectiveness of travel regulations and see if the stops in travel to the state did result in decreased number of cases statewide. And finally, we should be able to identify and trace hotspots of community spread and really look at those local clusters and identify where they are and whether or not those are dominated by community transmission. So thank you for listening. My colleague Claire Fraser will be presenting next week about genotyping SARS-CoV-2 and how we can observe the virus evolving. Feel free to contact us, and I want to give a special thanks to everybody in the Butler Lab, as well as Dr. Vivek Nirikar, who's and his tropical medicine clinical laboratory. They've been instrumental in getting the local testing facility set up out of JABSOM and running. So they're currently doing testing, and they're also currently starting to sequence Hawaii-specific samples of SARS-CoV-2. I would also like to thank you guys for listening and allowing me to present. And finally, if you want or know anyone who wants to support us, we do have a UH Foundation page that is open to donations to support research like this. Thank you so much.
Awesome. Thank you so much, Ethan. I learned so much, and it was in a very digestible format for someone, a lay person like me. I have a bunch of questions, but I want to give the rest of the participants here first crack at Ethan. So any questions for Ethan? Ethan, if you're an MS or a doctoral student, your defense is not going to be this easy. But
yeah, hey Ethan, that sounds great. It's outside of my scope. I'm data science, but I posted in the chat. Hey, and I'm at Maui College, UHMC, Debussy and I are. It'd be great to share your slides, or Thomas, if you're recording this with others that are bio people here that would be excited about this. If anyone wants to share, that'd be great.
Oh
For
yeah,
sure.
definitely. I sent my slides out. Feel free to use them.
I copied the Google, these are Google Slides, so I copied the link, a shareable link into the text. We can also share them, but we haven't done, I don't know if it will get messed up if we export it as PowerPoint. But Ethan, you're probably willing to distribute it, right?
Oh yes, definitely, 100%. This stuff is really exciting, and I want to be able to get it out there because I mean, this information is just so nuts. Because it shows how modern tools that look at things like genomics can really help contact tracing efforts. And the even cooler part about this is I know for a while, like the price per base pair for sequencing DNA was actually quite high, but with the just the influx of all of this next-gen sequencing technology, the price per base pair for sequencing DNA has dropped substantially. And you can sequence millions of bases for relatively low cost.
so I have a question on that. That was probably my biggest question, Ethan, in terms of if you go back to the previous slide and the applications or the implications for Hawaii. So for example, to make it super simple for someone like me to understand, if someone comes in, gets tested with a nasopharyngeal swab or a nasal swab for COVID for a PCR test, can you use that same sample? And then what does that cost? Because I don't understand I didn't understand anything about the base pairs, but how much would it cost to do that genetic sequencing or gene, whatever this great technology is? I apologize, this is way out of my depth, but if we were to try to do this for Hawaii, how much would it cost, a ballpark per person, to do this for the state?
So a lot of these reagents, it really comes down to the number of, so rewind real quick. You can use the same sample, yes. So if you get a sample from someone and they test as COVID-19 positive, you can use the same sample, take the viral RNA from that sample, and then do a complete workflow to get the entire genome. The pricing for this really depends on how many you are willing to do at once. So the caveat with next-gen sequencing technologies is that for a small number of samples, you're looking at pretty high prices. But as you scale it up, the price per sample drops significantly. We actually drafted up a mock budget of doing like this sort of analyses for samples from Hawaii. And if you look at about, I think, was it 40 samples or so, you're looking at about $100 per sample in the grand scheme of things. But if you double that to 100, it drops to like $50. And then if you keep increasing the scale, you can really drop that price per sample quite substantially.
That's very useful information. And hopefully,
And the other thing,
go ahead, Mark.
oh, go ahead, Thomas, sorry.
No, I was just gonna say, we have so many great collaborators and contributors here. Hopefully this can just, I see such huge potential for this for Hawaii, because we, so much of our economy, but also our transmission can depend on influx from returning residents and tourists. So if we can better understand, and we talked about this previously about the difficulty of contact tracing, right? So, using something that's a lot more time sensitive and is not as impacted by recall bias from contact tracing, this could be a potential next gen approach. So that was just my comment. Go ahead, Marguerite.
Yeah, I think the thing that's really exciting is that this work used to be much slower. So I mean, it comes from very backward looking types of work, but actually the workflows are much faster, and the sequencing is faster. the assembly and the analysis, the downstream analysis is faster. So that's what I think the future is going to be trying to use genomic tools for pandemic tracing. So we can almost basically process this in real time, that it becomes useful as a supplementary information for pandemic management. And that's kind of our hope. So it depends a lot on, you know, access to samples. Yeah, so we're still working on, we're getting some samples from Vivek's CLIA lab, but we are looking for more partners to obtain samples from. And then, yeah, and then we also are, we're applying for funding, you know, from lots of different sources.
Oh, I completely missed the chat questions. I'm so sorry. Let's see. Oh, question from Victoria. I've heard that the longer the virus evolves, the less virulent it will be. Do we have any reason to think that is true? So this is actually really interesting, and I know Claire will talk about this more next week. But what we've noticed, or what the research that's been done on the virus so far, heavily implies that there are two different viruses or lineages of the virus that dominate the populations at different times. And the key difference between these two types of virus is a mutation that occurs in the spike protein, which is what they use to attach to the cell. And Claire will definitely give more background or more information on this next time, but we do see evidence of different strains sweeping different populations. And as for the virus getting less virulent with time, I couldn't answer that, but.
Yeah, that's a very, very interesting question. And that's something that Claire, well, Claire is actually interested in researching the link between, you know, massive viral replication and spread and opportunities for evolution of the virus, and whether or not, you know, kind of the idea of like, is our response to the pandemic influencing the evolution of the virus? And looking at the correspondence between various, anyway, yeah, Claire will talk about this next week. But yeah.
Ethan, I think Francis has one more question, if you don't mind answering.
Oh yeah, of course. Does genotyping? Oh yeah. So does genotyping the virus from samples collected require special patient consent? To my understanding, and Dr. Barlow, please correct me if I don't understand this properly, but as long as there is no identifiable information attached to that sample, it should be okay. So as long as it's just a sample number or something with no identifiable information, we will still be able to use that information.
Yeah, yeah. We're, as a research lab, we're using de-identified samples, but from a clinician's perspective, you know, I don't know. You guys know how to deal with that. Like, if you want to actually convey the results back to the patient, that's beyond us.
And then a couple questions from Yosh as well.
Yeah. So average time between mutations. So it's about 2 per month is what they found. And with like the actual time between mutations is kind of rough right now, but 2 per month, 14-day transmission, or 14-day infection cycle, they're estimating based on those numbers. It's RNA. Oh yeah, since this is an RNA-based virus, I assume this has to be checked while the patient is infectious. Yes, so there are points where these sorts, so like for a nasopharyngeal swab, once the virus moves out of the upper respiratory tract and into the lower respiratory tract, grabbing virus from like nasopharyngeal swabs is a bit harder. So you have to catch it relatively early, at least to grab the virus from the nasopharyngeal swab.
Yeah, or from stored samples.
Yeah, or from stored samples.
Yeah.
Is ICE sequencing all positive samples? Oh, I actually don't know, but the UK, Britain, has made a very, very big effort to sequence as many SARS-CoV-2 positive patients as they can. And a little bit of background on them, they found some really, really cool, well, frightening stuff, but it is really interesting in terms of virus dispersal, because what they found is they kind of estimated that the number of long distance introduction events, long distance dispersal events from other countries was relatively low. But what they found after sequencing all the genomes that they can is that it's actually like thousands of times or 1,000 times higher than they previously thought. And this was very, very frightening because it wasn't just multiple countries, it was repeated introductions from multiple countries through time. Yeah, and I believe that paper is a preprint right now, but it's seriously full of great information.
Yeah, and I have no idea. I mean, if the DOH or anybody was storing samples through time, that would be so valuable to see, because then you could do things like observe how many lineages were present and how often they go extinct, you know, basically quelching the virus. But I just really have no idea.
Yeah, hopefully we can reach out to some of, you know, Dr. Desmond from DOH Labs, or I don't know, Dr. Berryman or Dr. Hankins, if you have any insight into storage, or, you know, whether or not DOH is looking into that. But, you know, as we all realize that this is a response that is going to stay with us for a while, and from this great presentation from Ethan, I'm looking forward to next week's presentation from Claire, you know, trying to use the cutting edge technology that we have to better inform our decisions, and not just our data, you know, our modeling, but just overall data points, decision making, data quality for our state moving forward. We have 5 minutes left, and this was such a great presentation and Q&A session, Ethan and everybody involved. I want to give back some time. I feel like we always go over, so want to end it a little early. Thank you everybody for participating. I love the new faces. Oh, go ahead, Marguerite.
Can I just say one thing? So even if we don't have every sample, right, it's still a very valuable thing, because we could still trace back the history of the samples that we do have. So it would just mean that there were parts of the family tree that were missing, but the rest that is inferred is still valid. So, you know, it certainly would be useful to know, for example, the history of the clusters and things like that. So I hope the state adopts this technology, because there's a lot of power from whatever it is we can manage to do. Thank you.
Thanks, Marguerite. This was great. We'll look forward to part 2 of this super interesting and hopefully impactful technology with Claire's presentation next week. Thank you everybody for joining, and stay safe. Have a great weekend, and we'll see you guys next week. Thank you, everybody.
Thank you.